Hi, Ten, and today we're going to be looking at another required practical, particularly osmosis, and we're going to be investigating the effect of the range of concentrations of salt or sugar solutions on the mass of plant tissue. Sounds a bit of a complicated one, but it's simply we're just looking at water moving, and I'm going to explain how that happens in a second. So, this again can be supported using your Caboodle textbook. And pages 16 to 17, there's a whole nice double page spread based on this, and use that. It has some really good diagrams. It shows you some hypertonic and hypotonic cells and what they look like, and it includes my favourite word, which is turgid. I think it's the best word in science. So, osmosis is the movement of water through a partially permeable membrane from a region of high concentration to a region of low concentration. Now, if it's water moving, hopefully that should ring bells, and you should be thinking, yes, osmosis. If there's no semi-permeable membrane, then it's just diffusion, and if it's moving in the opposite direction, then it's active transport. Hopefully that's been a bit of revision for you. So the diagram shows me my semi-permeable membrane. Now, cell membranes and cell walls are semi-permeable membranes. So that's important to remember. When they're talking semi-permeable membrane, they mean the outside of a cell. That's all they are talking about. Now, we've probably done this practical where you chopped up hundreds of bits of potato or carrot, and you can do it with an egg. Very exciting bit you can do with an egg as well. And you can see the water moving, not physically, but by changes in mass and shape. So the point of this practical, which I have often wondered, however, people have been doing it for years and they love it, and the exam board love it. So therefore, we are looking at the different effect of salt or sugar, not at the same time, guys. Salt or sugar, can be talking about either. How the water moves in and out of plant tissues. We don't really do this on animal tissues for a whole range of reasons. There's an ethical issue. People object to us chopping up small children. I haven't got a problem with it. But one of the things is that plant tissues, because they've got that nice thick cell wall, they keep their shape, and therefore it's much easier to measure them as tissues. Plant tissue is much more readily available, and you can buy a bag of potatoes for just under £2, which would last the whole of year 10 for this practical. I was only joking about chopping up small children. It's not really something I do. I'm vegetarian. Member of Greenpeace. Right. Health and safety. And you can think, what can go wrong with a potato? Well, there's an interesting one. So let's go through the equipment and then we'll go through the safety. Firstly, what we tend to do is set up a test tube rack with a range of boiling tubes with different concentrations of potato in each tube. The potatoes should be the same length or the same mass, and we need to weigh them and measure them before and after. Plant tissue, good one is potato. Another good one is carrot or swede, is also another really positive one. And we're going to use distilled water, and we're going to make sure we use distilled water this time so there's no impurities in that water that could affect our water movement. A sharp knife, also known as a scalpel. White tile for cutting on. Filter paper, tweezers, boiling tubes, measuring cylinders, ruler, balance, fancy word for scales, and stopwatch. So safety first. There are 3 things that we have to be really mindful in this experiment. Sharp knife and cuts, okay. Apple corers, again, for those of you who don't know what it is, it's like a cylindrical blade that you push down onto an apple and it cuts a perfect cylinder. And glassware. There's lots of water involved here, lots of moving glass around, and breakages do happen, so we do need to be careful of each of those. So, and just as a precautionary, because I have taught some of you, do not eat the plant tissues. Do not drink the water. Okay, just putting that out there. All right. So, can you just make a note about the risk assessments and sketch yourself a quick diagram? So, hypothesis. Now, some of these practicals, you don't really need a hypothesis, or they aren't really looking for that skill when they're assessing it in the exam. However, creating a hypothesis is really important, because otherwise we're just blindly doing experiments without thinking what they're about. So today we're going to be looking at what happens to cells when you put them in different concentrations of sugar, and how will the water move in and out. But a hypothesis is what you think will happen, and what you will think will happen based on your knowledge of what you know at the time. Now, in science, we often try and prove that hypotheses are either correct or incorrect. And it's not so bad if we get it wrong, because actually by ruling things out, we have learned information. And lots of great science discoveries have been made through getting things wrong. Okay? So there is no wrong, but it is all about your justification. And for those of you looking at a 6 or above, be really wary of this, okay? Be thinking, right, well, I think this, but why do you think it? Give a reason. Even if your reason is slightly off, they'll give you credit for it, so be aware. So I think the potato that is placed in distilled water, the mass of the potato will increase, and the mass will decrease when it's placed in a sugar solution. And we need to think about that. Why will that happen? Because the water will move because of osmosis. All right? Our independent variable, the variable that we have to change, is the concentration of the salt solutions. The dependent variable, the variable that we measure as a result of that change, is going to be the mass of potato pieces. Some people try and measure length. It really isn't worth doing, okay? It's really messy and you don't get very good results. The mass of potato is more accurate because you can calculate the change in mass, and we'll talk about that in a minute. Then control variables. So these have to remain the same throughout all of your repeats so that you can compare your results fairly. Be really careful, because actually they're going to want to know, do you understand why you're controlling this and why it's important? So the quantity of solution, the volume of solution that we use, time that is left in the solution, and then we're going to look at the method. Again, there are 11 steps to this method, so use the video that we did earlier about reducing that down. Right? You're going to cut your cylinders. You're going to trim them to the same length. So try and put these in maybe 3 to 5 words as you go down. Chimps can copy. Okay, guys, you need to think. You need to be looking at this method and go, well, I'm never going to remember that 150 words, but I have got a really good chance of being able to remember 33. And actually, when you're coming to remember 33, what you then do, you add a little picture at the end of each one, then you're going to actually remember more, because your brain will remember the picture and the words. So we do need to be retaining that information. Pause the slide, copy this down for me. Brilliant. So the last point you'll see is calculate and record the changes in mass. We need to work out the percentage change in mass, and that's the change in mass divided by the initial mass times 100. So just make sure that you've got that down. Now you can see the virtual experiment of somebody doing this, and it's awesome, on the YouTube clip, and I will paste that into the bottom of the link. Now, one of the other things that they mention here is the importance of accuracy. And the excess water is removed from each tissue sample before weighing. So one of the reasons that you might get a difference in weight that you weren't expecting is if, when you've taken it out of liquid, you then need to use that filter paper to blot off any excess water. And then cutting the length of the potato cylinder to each length, and potato skin, so make sure there is no potato skin left, because the potato skin will not be as permeable as those cell membranes. Just some fun facts, okay? All right. The last one that I think is really important is the temperature of the solutions, which Miss hasn't mentioned here, but on a couple of questions they have come up. And actually, the solubility, the salt within the solution, changes with temperature. So we've done this practical a couple of times with my class, where we've put the solutions in the fridge between lessons, and actually the salt has precipitated out of the solution, is as a residue, and therefore the solution is no longer at the concentration that we're expecting. So you can think about the temperature of the solutions and the impact on the solubility of the salt. So here we are, lovely results table. And what you need to do is calculate the percentage in change in mass across the middle. And these results can also be graphically represented. Now, obviously me and Mrs. Walker and Ms. Callis are working as a team. And what we've got here is a slightly different way of doing this. So, Miss has done something different to what I would have done. And personally, water shouldn't be there. The word water shouldn't be there because water has neither any salt or any sugar. I would prefer that to be 0. But Miss has put that there for reference, and I understand why she's done that. Also, lots of exams, especially on the higher paper, will move this x-axis up to where the 0 is across the middle. Right? We can plot our points like this. And we're going to then draw our line of best fit. And remember, our line of best fit can be a curve. Ignore Mr. Casey, because in science, a line of best fit can be a curve. Mr. Massey and Mr. Bethel will always say a line of best fit should be a nice straight line. Not in science, guys. It can be a curve. And that clearly is a curve and it's not a straight line. So, there's my lovely curve drawn by Mrs. Walker. And then we can make some conclusions. And this is a model answer for you. So the percentage change in mass of the potato decreased when they were left in higher concentration. And on the other hand, the mass went up when they were placed in distilled water. Think about how you could explain how these are. The idea of hypertonic, isotonic and hypotonic conditions. Okay, if you look at the diagrams underneath each of the cells, I think it might be a really good idea to copy those diagrams, and rather than blindly copy out this slide, surround each of those diagrams with notes to explain what is going on. This could be a model exam question, guys. It's that simple. And it has been summarised at the bottom of the screen for you. If not, if you're not feeling in the mood, pause the slide and just copy it. So, the skills gained, and they're going to be assessing in this practical. The idea of a hypothesis. We have a regard for accuracy in measuring reactants, okay? And identifying hazards, making and recording results, evaluate the method and suggest possible improvements for further investigation. Present results in appropriate graph. They quite like this one in an exam, guys. They often ask you to use the graph to calculate or to predict the result, a missing result. And use percentiles and calculate percentage gain and loss of mass in plant tissue. There are 2 useful links for this, and knock yourself out. I will add them to the bottom of the homework. I hope that's been helpful. Let me know. Alright then, take care, bye.